Good morning, everyone. Thank you for joining the webinar today, and we very much appreciate you taking an interest in the discussion that we are going to have. I'm very excited about it. My name is Mosam Shabela, and I'm currently the Acting Deputy Vice Chancellor for Research and Innovation at the University of KwaZulu-Natal. And I will be having a discussion with Dr. Nigel Garrett, who some of you may know, some of you may not know, but you are going to get to know going forward because of the amazing work that he's doing and he continues to do. Nigel is a specialist physician, focuses mostly on HIV and sexual health, originally from the UK, but has been in South Africa for nearly 10 years now, and working with CAPRISA, heading the HIV pathogenesis unit. He's got a number of clinical trials that he's heading, but most recently on our side of town, he's heading what we know as the Johnson & Johnson study, but he's also involved in the current rollout of the vaccine. We're very much excited to have the opportunity to hear more about this study. We will discuss the general vaccines around COVID-19, but also really get to closer and closely focus more on the rollout that's happening. But hopefully the heart of the discussions will be around the evidence around the Johnson & Johnson vaccine. I'm hoping that Nigel will speak for the next 25-30 minutes, and then we'll have plenty of time to engage in questions. Please put in your questions in the Q&A. We'll monitor them as we go, and then I'll bring them up for discussion later on. Otherwise, I already have a lot of questions for Nigel as well. Nigel, over to you.
Thank you. So Marianne, and thank you for that introduction, Prof. Musa. Marianne, I will now share my screen if you just give me the okay. Make sure you can see the screen.
Yes, it's appearing now.
You've got the full view?
We've got the full view. I'm going to switch off my video for now, and then you can carry on. Thank you.
Okay. Thank you, colleagues, for joining this webinar this morning. The title is COVID-19 Vaccines, A Path Forward for South Africa. It's a real pleasure to be presenting on this forum again. For those of you who don't know me, I'm the head of the HIV Pathogenesis and Vaccine Research Program at CAPRISA, also an honorary lecturer in the Department of Public Health at UKZN. Now, just a few disclosures. I am the site principal investigator of the COVID-19 Prevention Network, the CoVPN, that was formed about 8 months ago by the NIH in the US. I'm the sub-investigator on the Janssen J&J Ensemble trial that you may have heard of. And I'm also the co-PI on the Sisonke J&J vaccine implementation study rollout. So today I will talk about how we responded to the SARS-CoV-2 pandemic. I will briefly touch on a few SARS-CoV-2 vaccine concepts and also tell you a little bit more about some vaccine trials that were conducted in South Africa and internationally. I will then focus a little bit more on the Ensemble trial and the Sisonke Access Program that we just started about 8 days ago. Now, very briefly, this is my background. So when the epidemic hit us, we were mainly conducting HIV vaccine trials. One of them was the HVTN 705 Imbokodo study, which is still ongoing, which also uses the adenovirus 26 vector that is interestingly also being used in the Johnson & Johnson trial. So we had good relationships with the sponsor, with J&J, you know, that's probably also why that trial was conducted partially in South Africa. We also do lots of other work, including broadly neutralizing antibody work for HIV prevention. And as you know, in the COVID field now, it's not just the vaccines, there's also antibodies by Regeneron and Lilly that have been produced. So there's quite a lot of overlap and technology transfer from the HIV field to COVID, and probably coming back towards the HIV field. So just to remind all of us of the epidemic, some of you have seen this slide. So we know that it, essentially started becoming an issue in South Africa in March 2020. And then we experienced the first wave. In black, you can see the number of cases, and in gray, the number of hospitalizations. And then in red, you can see the number of deaths. Many of us thought that this would be over by July, August, but then we got this incredible second surge by about November time with a huge amount of hospitalizations, and also then following the death. And a lot of the deaths is obviously also excess deaths. So that's probably doubling the number of deaths, which is primarily due to just an overwhelming surge, which just completely overwhelms hospital and the healthcare sector. So the CoVPN is a network that was formed by the NIH, and we were part of that. It started working in about May 2020, and it was really the Operation Warp Speed, they termed it. We've got about more than 45 international clinical research sites, which all got together to start COVID, initially observational studies and then vaccine trials. So here you can just see how we set up our clinic. So we built a COVID clinic at our research site. And we also started doing outreach work with acutely infected patients. Here, we forged a good relationship with GPs in Phoenix, where we started a lot of our work. Here's a bit of a study schema of what we did. So the 5001 study was an acute infection study of people that had just been diagnosed. So they still had a virus detectable, and then the HVTN4/5 study that was more with convalescent patients. And we tried to enroll people that were asymptomatic, others that were symptomatic, non-hospitalized, and then also some severe cases. So that research has really helped a lot with understanding the pathogenesis of COVID, the viral dynamics, and the immune responses to the natural immune responses to the virus. Now I will switch now very briefly to a couple of, or 2 or 3 slides of maybe basic vaccinology. Not all of you may be basic scientists, so just to remind everyone very quickly, we're talking about the severe acute respiratory syndrome coronavirus 2. It's a single-stranded enveloped RNA virus. It's got 29,000 odd nucleotides. It's a mature SARS-CoV-2 virion, which has 4 structural proteins. So it's got that envelope protein, a membrane protein, a nucleocapsid, and importantly, that spike protein that everyone talks about. On the right, you can see a little bit how the virus infects our human cells. So it uses that spike protein, which attaches to the so-called ACE2 receptor, which then allows the virus to enter the cells. It reproduces in the cells, expels its viral code, and destroys the cells and then invades the next cell. In the second part of that panel, you can see a little bit the different immune responses. So usually it's innate immune cells that recognize the virus and then present the virus to these so-called T helper cells, which then signal to the cytotoxic T cells or CD8 cells to kill certain infected cells, and also signal to B cells to start forming antibodies. So that's basically the very basic immunology of the COVID infection. Now here you can see some of the vaccine concepts that have come our way. On the left in orange, you can see a concept where the virus is weakened or inactivated. And some of the Chinese vaccines, Sinopharm, for example, Sinovac, have used that approach. In the middle in green, you can see a replicating viral vector, which was used in the Merck measles concept, measles vector concept. Unfortunately, that program was halted because the initial responses were not strong enough. Then the non-replicating viral vectors, and the Johnson & Johnson vaccine is one of those examples, which use an adenovirus usually as a vector, and then an insert that codes the spike protein. And there's protein subunit vaccines, and Sanofi Pasteur are working on that. Unfortunately, their first phase 1/2 study showed low immune responses, and it was found that their protein concentration in the vaccine wasn't high enough. So they've just started another phase 2 study with a slightly higher dose. And then obviously the really exciting new kind of vaccine concept, which are these RNA and also DNA virus vaccine concepts, which you've all heard of, was very successful with the Pfizer and the Moderna vaccines. Many of us never thought that they would be as effective, in the high 90%, because you can imagine the RNA is just a single strand. It's very fragile, very heat sensitive. So they were delivered through a liposomal, like a lipid layer. So many of us didn't think this could be as effective, because there's obviously lots of proteolytic enzymes in the cells and so on who could potentially destroy these vaccines. Now, this is from a little while ago, this table, but you can see here the vaccine development pipeline. I'm sure it's now more than 250 concepts, but you can really see on the left that all the different concepts that I mentioned are all being explored. Most of them target try to generate the spike protein, which overall has been shown to be an extremely effective approach. Now, this table shows you some of these clinical trials that have been ongoing. I think we've got 3, 6, about 10 here. There may have been 1 or 2 additional ones now. It started off with the Pfizer vaccine reporting. It was a large trial of 44,000 in the US and Latin America, and also partially, Germany and China. Then the Moderna vaccine also reported. That was only done in the US. We all know the AstraZeneca story, which were multiple trials conducted in the UK, Brazil, and also South Africa. That vaccine uses the chimp adenovirus 5 vector. Then the Janssen trial, and I'm going to tell you a little bit more about that. The other trial that was conducted in South Africa was a Novavax trial, slightly lower numbers. And then there are other additional vaccines, the Russian vaccine called Sputnik, and 3 different Chinese vaccines. I know that I think Brazil has started rolling out Sputnik, and Russia, and obviously the Chinese vaccines are also ongoing. And here's just sorry, this is a little bit quite a busy slide. I just wanted to show you, this is an example of the Moderna vaccine safety profile. And then on the right, you can just see the neutralization responses that are commonly measured in these studies. So it's more of an example for what is measured in the early trials, the phase 1-2 trials. So on the left, you can see the systemic and local adverse reactions that we measure after vaccine administration. And in the middle column, you can see the responses after the first vaccination. And in the right column, the response after the second vaccination 3 weeks later. The gray bar means mild reactogenicity symptoms, blue is moderate, and yellow is severe. So maybe a severe fever would be a fever of about 40 degrees, for example. But overall, you can see that most of the adverse, the reactogenicity events are all mild. They're slightly, interestingly, with the mRNA vaccine stronger after the second vaccination, which probably indicates that the immune system was properly primed with the first vaccination. And then on the right, you can see this neutralization panel. So the higher the little bars, the more of an antibody response you see in the trials. And you can see that these responses last here in this example for about 2 months at least. There's still a bit of a question mark about the durability of all of these vaccines, but I think we're getting more and more implementation data now, and we will find out very soon how long, or what our vaccination sequences will have to be, whether we need annual vaccines or whether a single dose is fine. Now I'm going to tell you a little bit more about this Ensemble trial that we've just pretty much completed. There was a multi-center phase 3 double-blind 1-to-1 randomized trial. This is a single-shot vaccination regimen. So only 1 dose of the vaccine was given compared to most of the other studies. The vaccine was an adenovirus 26 SARS-CoV-2 vaccine. The adenovirus 26 platform has already been used for many other vaccines, including Ebola successfully, respiratory syncytial virus, for Zika, and also we're using it in the HIV vaccine field. So we have a lot of experience in our clinic with that vaccine platform, and that's why we were very excited to be part of this trial. It's also stable at 2 to 8 degrees Celsius for 28 days, which helps with the cold chain compared to, for example, the mRNA vaccines that need to be stored at this very low temperature to remain stable. Here you can just see a schedule of evaluation of the participants in the trial. We enrolled 7,000 participants in South Africa. So people got their vaccination, were screened, and then got their vaccination on day 1. They then returned on day 28 for blood results blood tests. And in between, we did COVID-19 signs and symptoms surveillance. That was all done electronically. So as soon as a participant experienced any symptoms, they would use their smartphone. If they didn't have a smartphone, we would provide a smartphone, and immediately that message would be relayed to the vaccine team, the research team, and we would then follow up with home visits and frequent nasal swab testing. So yeah, so the interim analysis was conducted quite early, so I'll get to that in a minute. We're still following up everyone. The the FDA are reviewing all the data now. So if they license this product, then we hope to provide the vaccine to all the placebo participants as well. So in the trial overall, we enrolled 44,325 people. There was a good age distribution. More than about 4.5% were over 75. Good gender, even balanced gender, and good regional distribution. And also it included many people with comorbidities. You can see here, for example, 30%, almost 30% were obese, about 7% had diabetes, 10% were hypertensive. And also we included about 3% HIV positive volunteers. Now the overall results, you may have heard this already in the media. The vaccine was 72% effective 28 days after the vaccination, 66% effective overall against moderate and severe disease, and 85% effective against preventing severe disease beyond 28 days after the vaccination. It completely protected against related hospitalizations and death. And importantly, it was also protective, slightly less protective, but 57% protective against the circulating variant at the time in South Africa, the famous 501YV2 variant lineage. Now here, I just wanted to show you again these neutralization titers. So this is from the Johnson & Johnson trial, and it shows even after one single shot vaccine, you can see on the x-axis, the responses in green were quite high up to about 3 months out. So we will now have to monitor longer-term immunogenicity and antibody responses with these vaccines. So overall, in terms of safety, the vaccine had an acceptable safety profile. It had just the common reactogenicity events that you would expect. There weren't really any safety signals. And importantly, there were no cases of anaphylaxis in this trial, which was a little bit of an initial worrying signal in the mRNA trials, but that is no longer really an issue amongst the mRNA trials either. And obviously, it's already the UP26 vector has been given to more than 200,000 people previously. So we're pretty confident that this is a safe vaccine. Now, J&J have applied for emergency use and have submitted a request for an EUA, which is an emergency use authorization at the FDA. Now, we're quite excited. The decision is due either today or tomorrow. Now, they're also submitting EUAs to other regulators, and SAPRA has already been informed. There's a rolling submission to SAPRA in South Africa, partially due to the Sisonke program that we started. Now, just to put this in context, there's not just the Johnson & Johnson trial, there are many trials. Some of these vaccines have been highly efficacious. If you look at the Pfizer and the Moderna one, they were kind of the front runners with the 95% efficacy. The others were also doing well. So a rough guide is if any vaccine that's more than 50% effective is probably a vaccine you can roll out according to WHO recommendations. The AstraZeneca vaccine, there's a large trial still ongoing in the US, but that has also shown efficacy. Unfortunately, as you know, it didn't work as well against the variant virus, and we'll get to that in a minute. Obviously, it's important also to look at prevention of hospitalization and severe disease. And here you can see a rough idea. The Johnson & Johnson one was about 85% effective. Moderna and Sputnik viruses, vaccines, reached almost 100%. And in the Novavax and AstraZeneca trials so far, we had slightly too few outcomes to draw strong conclusions. Now, in terms of the variant, we were part of, so some of our colleagues, Tulio de Oliveira and Alex Segal and Penny Moore from South Africa were involved in some of these studies, looking at serovirion and live virus lab assays to see whether the neutralization activity of the vaccine was reduced with the variant virus. And you can see across the board there was a reduction, especially we were extremely concerned about the AstraZeneca vaccine, and you all heard that that vaccine rollout was then put on hold for now. It also reduced the efficacy of the Novavax vaccine in South Africa. What is important to know about the Johnson & Johnson vaccine, it was actually conducted during the spread of the variant, and it showed 57% effectiveness. I think 98% of the breakthrough of the circulating virus at the time was the variant, and it was highly effective against severe disease. Now, just looking at an example of a vaccine rollout, it seems like Israel are doing very well, and the impact of a potential vaccine on the epidemic curves. So they have now vaccinated, I think, more than 80% of their 60-year-olds and over in the country. And you can see, it looks like, I mean, we know that there's an epidemic curve, of course, as well, but there's looks like a definite decline now in number of cases after this rapid rollout of vaccines has started in the country. And we're hearing very similar news from, I think, Scotland also are reporting that these vaccines are as effective in the real world as they are in these clinical trials. Now, this is the problem we faced a few days ago still, that although their vaccine being rolled out in the US, South America, and Europe, mainly the map across Africa is still relatively empty. So this is the sequence of events that I've been involved in the last 2 weeks, and it's been a real whirlwind experience. So we started the Ensemble trial in South Africa on the 7th of November. The interim results already were released by Johnson & Johnson on the 29th of January, which is only about 2 to 3 months later. I then received a phone call from Professor Glenda Gray, who all of you know by now, whether I could help with this Sisonke access program. And me and others then, including Prof. Amina Goga and Linda Gelbecker, wrote a protocol together in 24 hours. We received BREC approval for the study about 2 or 3 days later. The protocol was submitted to SAPRA, and all approvals were in place essentially a week later. And we then managed to get the product to be flown in from Belgium on the 17th of February last week. And on the same day, we vaccinated the first healthcare workers here in KwaZulu-Natal at Prince Mashini Hospital and Albert Luthuli Hospital. Now, I'm going to try to tell you a little bit more about the Sisonke program, about 10 more slides. It's an open label, single arm phase 3 implementation study to monitor the effectiveness of the single dose At26-CoV-2 vaccine from Johnson & Johnson among healthcare workers in South Africa. It's a collaboration with the Department of Health and the South African MRC. The purpose of the study is to monitor the effectiveness of the single dose vaccine among healthcare workers as compared to the general unvaccinated population in South Africa. It's an open label, single arm phase 3 vaccine implementation study. We're aiming to enroll healthcare workers that are age 18 and above in the public and private sector, including GPs, up to 500,000 people. And the monitoring phase surveillance will continue for about 2 years post-vaccination. The primary objective of the study is to assess the effectiveness of At26, the vaccine, on severe COVID, hospitalizations and deaths in healthcare workers as compared to the general unvaccinated population in South Africa. And then we have additional secondary objectives, like looking at the incidence of SARS-CoV-2 infections among this population, to monitor the genetic diversity of the breakthrough infection, to keep an eye on viral evolution, to estimate the vaccine uptake amongst healthcare workers, and overall to conduct pharmacovigilance to monitor safety and any unexpected adverse events of the vaccine administration. Now, the challenge of this program is that the vaccine was not licensed at the stage. We knew it was safe and efficacious and relatively easy to roll out. So we formed this partnership, and the vaccines arrived and then were distributed to the 18 research centers where we conducted the Ensemble trial. The Ensemble research teams became the so-called guardians of the vaccine, which at this stage is still an investigational product. And we've been delivering the vaccine to the vaccination centers. So just to show you what we did is in KwaZulu-Natal, we have 6 research sites and research pharmacies, 3 with the MRC in Boters Hill, Chatsworth and Tongaat, 2 with Caprisa in Vulindlela and eThekwini, and then 1 in Ladysmith. So we basically mapped out the research sites with concentric circles in terms of distance and tried to establish how many hospitals, and public and private, were in the vicinity of these research sites. For the first round, we then picked essentially the 2 hospitals that were highest in terms of healthcare worker density in the surrounding area. And looked also at COVID rates at these hospitals and feasibility of delivering the vaccines to those sites. Now they have these 5 steps for the healthcare workers to remember. You need to register on the system called EVDS. I'm going to talk about that in a second. You need to respond to an SMS invite. Then you need to provide consent, which is an e-consent we built into the system. Then you receive your vaccination voucher, which is essentially your ticket for the vaccination. And then you can attend the vaccination center for administration. The healthcare managers needed to know that, we will deliver frequent deliveries of about 80,000 doses every 2 weeks, up to a total of 500,000. We were focusing on large hospitals in the pilot study the first 2 weeks. We will then help with the cold chain and draw up the doses, the research sites, and then the vaccinators of the DOH will actually administer the vaccine. This is just to show you quickly the portal so that you don't forget to register. And here you can see a schematic flow of participants through the system, the EVDS system. So you've got your healthcare worker that registers, receives the first SMS, then receives a second SMS whether they want to join this Sisonke access study. We then inserted a consent step. So there's a link to a website that leads you through a patient information sheet and then electronic consent. Once you've consented, you will then receive a voucher SMS, which can then be taken to the vaccination center. And eligibility is confirmed there by an administrator, and then you can move to the vaccinator stage and receive your vaccine. And you then receive your confirmation SMS with a voucher, with a vaccination code proving that you had your vaccination. Now, we also built in several safety nets. So we created a clinical safety desk, which takes phone calls with a toll-free number. We also created a protocol safety review team and a safety monitoring committee. So anyone can call into the safety desk. And obviously the vaccination sites and hospitals can also report AEs. And then we compile reports every 2 weeks to SAPRA working very closely with their pharmacovigilance team there. We're using exactly the same forms. We've just redesigned things a little bit. So we have a simple REDCap database that is also sent to participants once they've received their vaccination confirmation. And that allows us to create these guidance sheets as well. You know, we're looking at AEs in real time. So we know roughly what kind of symptoms participants are experiencing. I mean, one important part to say is it's you know, it's very normal to get some reactions after the vaccine. Obviously, some local, you know, pain, swelling, and redness, but also some fevers, chills, and headaches, tiredness. Perhaps some people mentioned that they had some insomnia, a little bit of lack of sleep for one night, joint aches and nausea, but usually these symptoms subside within about 48 hours. Now, this is where we were yesterday. So we started vaccinating and the launch was on Wednesday last week. So far, we have vaccinated 51,000 healthcare workers across the country, approximately 7,500 in KwaZulu-Natal. And we're vaccinating about 12,000 a day now. In KwaZulu-Natal, we're working very closely with the province now, the Department of Health. They have been involved in the selection of the next batch of hospitals. So we about to complete Albert Luthuli and Prince Mashini, and the next round will include Stanger Hospital, Edendale Hospital near Pietermaritzburg, and also Madadeni Hospital in Newcastle. So we're ensuring that all districts are also served. We're also hoping to start a center next week at St. Augustine's Hospital in Durban to continue to work with the private sector as well and the GPs. So in summary, the pandemic has affected all of us. I think especially in November, December, many people were affected by the pandemic, and economically everyone had a huge burden to deal with, and it's ongoing. The research has been conducted at breakneck speed. As you could see, we only discovered really the virus early last year, and it's quite phenomenal that we are rolling out a vaccine within the year to our population. The emergence of the new variant has complicated the choice of vaccines, and the discussion on that is ongoing. You've all heard about the AstraZeneca delay. And then South Africa has made a huge contribution to COVID-19 vaccine research, not just the J&J Ensemble trial, but also the Novavax trial and the AstraZeneca trial. The Sisonke program is one way we felt to kickstart the vaccination program in South Africa. We really felt it was time to start, and it has generated a lot of momentum, we feel, also a lot of training opportunities for ourselves, but also for the DOH staff. So we hope I have convinced you that this vaccine is safe, and hopefully we hear more today from the FDA. So I urge you all to register on the program. Thank you very much, and I'll hand back to Prof. Musa.
Thank you so much, Nigel. That was amazing. And you can stop sharing the slides, and then let's have a little bit of a discussion. And I think that looking at the questions so far, it does look like we just need to go back to some of the basic explanations, and I'm really glad that you provided an update on the Sisonke study as well.
Sorry, so, I'm just trying to get rid of my slide.
Stop sharing, yeah. Should be at the top where you stop sharing, but I'm sure even Marianne can assist you there. Anyway,
can you assist me, Marianne? Thank you.
Yeah, excellent. Yeah. So let's start firstly with some of the basic explanations. So The first thing is, as you said in your closing remarks, that we've got a virus that is relatively new, but we already have a vaccine for it. Can you just explain how it's possible that we are still trying to understand a virus, and we have learned now that the virus is evolving, and yet we have already established the vaccine, we've already made the vaccine. How's that possible? Can you just, in simple terms, explain that briefly?
Thank you, Prof. Musa. I think we're all still learning and trying to work this out. Now, we've learned a lot about, the traditional way of running these vaccine trials is you do your very early, very small number phase 1 study. Well, essentially, first you do animal studies. Then you go do a very small phase 1 study. Then you do a phase 2 study, which is like an extended safety study. And then you do your efficacy trial. So in this incident, they did the animal studies in record time. I think they managed to obviously isolate the virus in January last year. And then the laboratories with this new technology were able to sequence the virus so fast. That's why we had a leap of lab technology. And I think by March time, these studies already started. Then we had very quick phase 1 studies. So what happened, I think, is that a lot of these vaccine platforms, you call it like a platform, were already available. So what the manufacturers did is they just...
So basically, when you say platform, you mean like the different technologies you can use to design a vaccine. Like when you say a vector vaccine or mRNA vaccine. Okay.
Yeah. So you usually have your vector, which is just a shell to deliver. It's like a transport vehicle to deliver the little piece of the virus to the immune system. So if you... we already had all that in place, and what they said is, okay, we'll stop the production line and we'll just add a little COVID insert to the vector. So we're basically changing the goods in the truck. We're not delivering bananas, we're delivering apples. And that was very quick. And then the other thing we learned as clinical researchers, we can actually run trials much, much quicker than we thought. So in HIV, these trials are ongoing for 2 years. But if we're looking at the numbers, so if you have 40,000 people in a trial, you can get your primary outcome much, much quicker. You'll accumulate infections much quicker. And these COVID surges, so the epidemic, also really hit us so hard that suddenly in South Africa, when we were running the study, we had daily infections in the trial to report because almost, there were so many infections in the community that helped. The other thing I think what helped is that these vaccines were so efficacious. So if you have a 90% efficacious vaccine, that means you can say that there's an effect much earlier than if you have a 20% effective vaccine. Okay. So the difference between the placebo arm and the intervention arm was so huge that it became very quickly obvious that the vaccines are effective. So all that allowed us to save time. We still went through all the safety steps, but that allowed us the time. And then I think obviously our efforts then also helped us to get the vaccine into the country quickly.
Okay, so we can then conclude that we trusted our initial understanding of the virus, we trusted the technology that was available, and we were able to adapt it. And then now we can also say that, we can have confidence and trust the process that was followed in basically testing these vaccines. But now the virus keeps changing. How does that then affect the vaccines and the technology of the vaccines? Like, can we still now trust the vaccines if we know that the virus is changing?
Yeah, thank you, Musa. I think that the variant has caused a lot of new questions for everyone. I think the scientists are working on it. our own Professor Tulio de Oliveira is still sequencing the virus as we speak.
Yeah.
So I think the key will be ongoing surveillance. So when we collect these nasal swabs, they will be stored by the NHLS or, the National Institute of Communicable Diseases. I think it is important that we keep sequencing the virus to keep an eye on any changes and mutations, what they call mutations. And
right.
Now that we're running this program, what we added to the program is that every infection we will sequence. So we know through the EVDS system who registers and who gets the vaccine. So we can then link that with the NICD data, the National
Institute of Communicable Diseases,
data, and then sequence the viruses and see, has it changed again, or maybe even has it changed because we're now putting pressure on it with the vaccine.
Exactly, yeah. Okay, so that's helpful. Now, what you are saying is that not knowing enough about the virus should not stop us, or the fact that the virus is changing should not stop us from putting in place interventions. We should go ahead based on what we know, and then we can just adapt as we go along by making sure that we are monitoring the situation, we are surveying the situation, and then we keep adapting. Is that a fair conclusion to that question?
Yeah, I think we have been kind of caught out by the virus when it hit us, the first wave. We were a little bit surprised with the second wave. So I feel the virus has been ahead of us for quite a bit now, and that's why we saw such a traumatic impact on deaths and hospitalizations and so on. So I think what we're trying to do, we know this vaccine works. So the quicker we can now roll it out, the more of an impact we think we can have. If we wait 6 months, the virus may have mutated again, and then it may not be as effective. So we essentially, once you have an effective vaccine, I think it's very important to roll it out quickly so that we can have the biggest impact on the epidemic.
That makes sense. So we just need to make sure that we get the vaccine as quickly as possible so that we don't give the virus the chance to keep growing, and therefore giving it the space to keep evolving. So let me then ask you the interpretation of efficacy. The attendees want to know, when you say 57%, when you say 85%, how do we interpret that in simple terms? What does 85% efficacious actually mean, or 57% in simple terms?
Okay, good question. So essentially, it's a numbers game. And what we do is, at a certain predetermined time point, let's say in this study, it was 28 days after the injection. We count the number. So we've got equal numbers in each arm, in the placebo arm, and the dummy arm, and the vaccine arm. We then essentially count the number of cases of COVID that we experienced in the placebo arm versus the vaccine arm. And then it's essentially a simple proportion. So we saw 85% less hospitalizations related to COVID in the vaccine arm amongst participants that were randomized to the placebo arm. The vaccine arm. So I'm not sure whether that makes sense, but it's obviously there's also a time component to it. It's not just a simple, So some of you may know what I've seen Kaplan-Meier graphs, but you're actually looking at the incidence of new cases, and that is 85% less in the COVID vaccine arm.
Okay, so then does it mean then I will be protected 85% of the time, or does it mean out of 10 people, 8 to 9 people would be protected? What does that mean for me?
Right. So, most importantly, I think for the individual is severe disease. If someone gets a common cold, like, symptoms, people usually cope with that. They may lose 1 or 2 days at work. But if we can prevent severe disease or death by 85%, that is a huge impact for the individual, definitely.
Okay.
Now we don't yet know exactly how well this vaccine prevents transmission. Now that is important for public health.
Yeah.
Okay. But there is now some evidence that all these vaccines are having a big effect on also transmission. So that would mean then you get less individuals that are actually infected. So you then have an even bigger impact also on severe disease, because people that are not infected will not get a severe disease as well.
Okay, so is it then fair to say that the vaccine does not prevent infection, but it will help to fight infection and prevent disease or severe disease? But because it does that, then it helps to prevent the spread to the next person, generally in the population. And that's how it basically disrupts transmission. Is that fair to say?
Yeah, I think you summarized it nicely there.
Okay.
So just, Musa, maybe just one thing to add. What we're seeing now, with the variant research that's been done on the AstraZeneca vaccine, where suddenly what they call the efficacy went down to maybe 20% or even just 10%.
Yeah. Right.
At that stage, people will think, oh, I need to take this vaccine and I'm only maybe have a chance of being protected by 10%. That is not enough incentive for me to take this vaccine. So this is now what's happening in Europe, that people get called to the vaccination center and say the vaccine is ready, but we cannot guarantee which vaccine you will get. Then they say, okay, you've actually, we don't have any Pfizers anymore. You will get the AstraZeneca.
AstraZeneca.
Some of our European friends get up and walk straight back out of this vaccination center because they just feel maybe the vaccine is not strong enough. But I think we know that this vaccine, once you're at a 50% level, the impact a vaccine can do on an epidemic is quite remarkable. So I think the key is that as many people as possible buy into this, so that we don't just vaccinate a few, but as many as possible, and then we start protecting each other that way as well.
Okay, so I'm going to ask you the last question, and then I think there's a few people who want to ask questions. And the question that was raised here, it's about long-term effects of the vaccine. So one aspect of it is that when you stop the trial, then you're giving the vaccines to the people in the placebo group, which is fair. You need to do that. But now, if you no longer have a control group, how are you going to monitor the effects of the vaccine over that 2-year period, which it's critical to establish the long-term effects?
Now, this is the very important point, and it's been debated amongst us scientists for the last 3 to 4 months since the Moderna vaccine was licensed. Now, we feel the ideal study design would be what they call a blinded crossover. So a blinded crossover would be you keep the volunteers blinded in the study, but you ensure you basically tell everyone you would have got the vaccine either at the beginning or now. Let's say we're 6 months down the line. So what you do is you give all the placebo arm people the vaccine, and all the active, so the vaccine arm people get the placebo. But you're going to do it blinded to the clinicians and blinded to the volunteers. So that way the volunteers know that they received the vaccine, but they don't know whether it was in the first wave, the first round or the second round. Now that would allow us then to still monitor durability of these vaccines a little bit, and also maybe a little bit more objectively monitor side effects.
So that's good. So let's take 3 quick hands. Professor Sine, can you go first? And then I will also invite the next 2 people, and then Nigel, you'll answer the 3 questions together. Go ahead, Sine.
Sinead's dropped her hand. I think she didn't have a question.
Oh.
Maybe we can go to Kine.
Okay, Kine. You can unmute. Just unmute again. Marianne, if you can assist to unmute Kine, that'll be great. Maybe we can go to Dharma and then we can come back to Kine.
Dharma, please unmute.
Okay, I think they're being shy, Nigel. I don't know what you did to them there, but there's the last question in the chat. Let me ask you this last question. It's about the fact that it looks like we are not going to get ahead of the virus with the vaccines in the short term, given that we are anticipating the third wave. Hopefully, we can prevent a fourth wave. Now, what are our options? Should we be trying other avenues? Others are asking, should we be trying ivermectin? Should we be trying other vaccines? What are our options in your view?
Yeah. So I think we know that healthcare workers have about a 3 to 4 times risk of acquiring COVID-19. So that is why we went now as a first step into that population to offer them the vaccine. I think obviously then you would have to look at vulnerable groups, older volunteers and people with comorbidities. So I think if we could try to target with the limited vaccine that we have at the moment, the most vulnerable in the population who are most likely to have morbidity and maybe die even from COVID. I think that is our responsibility now. Now I'm not part of the ministerial advisory committee or the the minister's office, so I don't exactly know where the negotiations with the manufacturers got to. I've heard that the Johnson & Johnson vaccine, that there may be an arrangement that up to 9 million vaccines may become available. I know that the government are also negotiating, I think, with Pfizer. Now the AstraZeneca is a real shame because it was a cheap vaccine that could have been manufactured very quickly. So we may not be able to use that. Now, in terms of other things, I think in this situation, we know the virus, we know the vaccines work, and we were lucky enough to pick vaccines that make the spike protein, and they work. So I think we just got to try to get vaccines out. My knowledge of this comes up a lot in discussions, the ivermectin. I think it's a personal choice now for individuals. The evidence, I think, will grow on ivermectin. There are some ongoing trials that are a little bit larger than the ones that have reported. The confidence intervals in the ones that were reported were very broad, so very difficult to make very conclusive conclusions. But so I don't think there's much else at the moment, apart from obviously the non-pharmaceutical interventions that we've all implemented already, the distancing, sanitizing, and mask wearing. And even, I mean, we do recommend that for people who received the vaccine as well, to carry on sticking to those procedures. Yeah.
All right. So that will be the end of our webinar. Thank you so much, Nigel, for the update. And then when you do have, after you've done more work on this Sonke, we would really like to invite you back to give us further updates. And we really appreciate the time you've taken to share this with us. And we know that we are keeping you away from more important things to do. So we really appreciate this. And thanks to the College of Health Sciences for organizing this webinar and all the attendees. Thank you so much, much appreciated.
Thank you, pleasure.
Thank you.